students, you are listening to podcast Botany for NEET by S. Ramakrishna. In this podcast, the topic photorespiration. In case of normal respiration occurs in light as well as dark, CO2 released and glucose it is oxidized, CO2 release occurs in mitochondria. In photorespiration, it is a light induced CO2 release. The CO2 release occurs in mitochondria. And this photorespiration involvement of the three organelles, chloroplast, peroxisome, mitochondria. Before that, you have to know about the Rubisco and RUBP. Rubisco is a bisubstrate enzyme. It's the main substrate, it is RUBP. Co-substrates are carbon dioxide and oxygen. So Rubisco bind with CO2 or O2, CO2 and O2. That should not mean that one Rubisco enzyme is not going to bind with CO2 and O2. It occurs at tissue level. Some Rubisco molecule bind with O2, some Rubisco molecule bind with variety CO2. This happens in case of the C3 plant. If CO2 and O2 are nearly equal, Rubisco act as carboxylase only. Because Rubisco problem is not having specificity but having affinity. It do not recognize the CO2 or O2. Both are gases in nature and small, but having affinity for carbon dioxide as well as oxygen. In nearly equal concentration of CO2 and O2, Rubisco mainly act as carboxylase. If they are not equal, what happens? Their binding is going to be competitive. At high CO2 concentration, Rubisco act as carboxylase, RUBP plus CO2, Rubisco act as carboxylase, two molecules of phosphoglyceric acid, that is two molecules of 3-PGA formed. Next, when it is not so, Rubisco act as oxygenase, that is RUBP plus O2, Rubisco act as oxygenase, a molecule of 3-phosphoglyceric acid, that is a 3-carbon compound, and a molecule of phosphoglycolic acid, it is a 2-carbon compound. So there is a photosynthetically fixed carbon will be degraded, some carbon going waste as carbon dioxide. So carbon dioxide release occurs in mitochondria. There is neither synthesis of ATP nor formation of any NADPH. Rather, ATP is utilized in order to release the CO2 in mitochondria. In C3 plant, as some amount of O2 bind to the Rubisco, photosynthesis is reduced. Productivity and yields are reduced in case of C3 compared to C4. In case of C4, at a Rubisco enzyme site, much CO2 is available because of decarboxylation from C4 acids, that is malic acid. So Rubisco minimizes its activity as oxygenase. So no photorespiration detected in case of C4 plant. So C4 plant are said to be biologically advanced. Productivity and yields are high in case of C4. Why? Because PEPcase, phosphoenol pyruvic carboxylase, having affinity with only carbon dioxide. That is one advantage. Rubisco minimizes its activity as oxygenase. No photorespiration detected. Further, C4 plant tolerate to temperatures like 30 to 40 degrees and respond to high lighting intensity. That's why C4 plants are biologically advanced. In this key point, number of organelles in photorespiration 3. One, it is peroxisome, single membrane, other are two membrane bound organelles. What is the substrate for Rubisco? RUBP is the main substrate. Co-substrates are CO2 and O2. So when it acts as oxygenase, 2-carbon compound formed, it is phosphoglycolic acid. And physiological significance of photorespiration not yet known so far.